Good afternoon. I'm Dr. Brian Shapiro, a Market Segment Manager at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series. It's entitled Optimize Your 3D Culture Workflow with Organoid Growth Kits, presented by Mr. Steve Budd. Dr. James Clinton and Mr. Stefan Friend appear in the tutorial segment of this presentation. Dr. Clinton then joins us as a panelist during the Q&A session. Mr. Budd is a product specialist at ATCC, while Dr. Clinton is a lead scientist. In this presentation, Mr. Budd will give some background on the Human Cancer Models Initiative, describing organoids and other advanced models. Then, Dr. Clinton will provide instruction on how to produce growth media using ATCC Organoid Growth Kits, while Mr. Friend demonstrates. After he closes, Mr. Budd and Dr. Clinton will join us for a lively Q&A session that addresses your most technical challenges in organoid growth. If you have any questions for our speakers, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome Steve Budd.
Thanks for the introduction, Brian. Founded in 1925, ATCC is a nonprofit organization with headquarters in Manassas, Virginia, and an R&D and service center in Gaithersburg, Maryland. We're an innovative R&D company featuring gene editing, differentiated stem cells, and advanced models such as organoids, which is the topic of today's presentation. During this presentation, we will give an overview of the Human Cancer Model Initiative and give descriptions of the models we offer. We'll also show what support is offered for these models in the way of clinical and genomic information, as well as organoid culture support. This will include our new Organoid Growth Kits designed to simplify the complete organoid growth medium workflow. We're also going to play a video demonstrating the media preparation using these kits. There is an unmet need for better preclinical cancer models. Why are existing cell lines used for preclinical studies insufficient? Many models are available in the way of classical cell lines, but are lacking specific tissues such as liver and pancreas, where these models are underrepresented in research and clinical studies. Many cells lack historical data, both in the way of patient demographics and clinical outcome. Also, many cell lines have been in culture for so long it can be hard to determine how representative they are to the original models. Current cell line models, consisting of single cells, do not imitate the complexity of cancer diseases. They lack the genetic diversity and different cell types that exist in real cancer tumors. In short, cancer cell lines are not true biological representatives of tumors. They lack biological relevance. More advanced models such as patient-derived organoids can help address these deficiencies. As an overview of the HCMI program, the key players are divided between the founders, model developers, and distributor. The founders who fund the program include the National Cancer Institute, Cancer Research UK, the Hubert Organoid Technology Foundation, and the Wellcome Sanger Institute. The model developers are the ones that generate the models from the patient-derived biopsies and consists of the Broad Institute, Cold Spring Harbor Laboratory, Wellcome Sanger Institute, the Hubert Organoid Technology Foundation, University of Verona, Hubert Institute, Stanford University, and Weill Cornell Medical College. The models are then sent to ATCC for final distribution. ATCC is currently the sole distributor of the HCMI models. This is a workflow that describes how the models are generated and distributed in more detail. The model developers work with clinical sites where biopsies and clinical data is taken. Biopsies will be sent for both sequencing and to the model developers for generation of the in vitro models. The models themselves will be sent to ATCC for expansion, quality control testing, and distribution. The subset of models that have been expanded at ATCC will then be sent back for resequencing. ATCC will continue to expand quality control test models before distribution to the biomedical community. One important aspect of the uniqueness of these models is the level of characterization that they undergo. This includes molecular and clinical data. The desire is to make available whole genome sequencing and whole exome sequencing. As previously stated, this sequencing is done on the original biopsies. A significant amount of patient data is also collected, which includes the diagnosis, patient background demographics, and the treatment that patients received, as well as the resultant outcome. A little later on in the presentation, I will show you where this data can be accessed. The HCMI models consist of different technologies. This presentation is going to focus on organoids and organoid culture techniques. But in addition, we do have various other model types, such as large assortments of neurospheres, which are 3D spheroid glioblastoma models, and other more standard 2D models that may represent more rare cancer types. So specifically, what is an HCMI organoid? You need to note that organoids can be made in various ways, such as through normal primary tissue or through iPSCs. Organoids in general are self-organizing microtissues that grow in a 3D extracellular matrix. But these HCMI models are unique. As I previously mentioned, they're all patient-derived from biopsy tissue. They consist of multiple cell types and exhibit cellular polarization. This allows for in vivo architectural features. As an example, in this image, you can see the lumen formed, which is a common feature for these organoid models. Similar to classical cell lines, they exhibit long-term expansion. They can undergo multiple passages while being shown to be phenotypically and genetically stable. So here I'm going to touch on the organoid culture technology. The technology originally came from Hans Cleaver's lab at the Urich Institute in the Netherlands. As it applies to human organoids, it relies on an advanced medium that can include conditioned media from wet and responding cell lines, recombinant proteins, and small molecules. This diagram is an overview of the culture process. To begin, suspended cells and fragments are dropped into the extracellular matrix. They are incubated at 37 degrees, where the matrix polymerizes into a gel, forming a dome structure. Liquid media is then overlaid on the dome. The cell structures then develop and enlarge into these complete organoids. The bright field image on the right is an example of a real model, so you can see what they actually look like. In this image, you can see the lumen, which gives this organoid a balloon-like appearance. To passage the models, the organoids in ECM is collected. The ECM is removed and organoids are enzymatically disassociated. At this point, they can be replated and expanded again. For the next few slides, I'll focus on the resources available to access supporting information, such as the clinical and sequencing data, as well as information and support ATCC offers. Here we have the HCMI searchable catalog managed by NCI. It integrates some clinical information and allows you to search from the model based on characteristics of your choice. You can search for the models of interest based on a number of filters, such as primary tumor site, model type, diagnosis, stage, grade, histological type, as well as gender, age, and ethnicity. One thing to emphasize is that this link directly to the ATCC product page where the models can be purchased. So if you go to this catalog and narrow your search until you find the model you want, you can be directed to its location on ATCC's website for purchase. The Genomic Data Common, or GDC, is another NCI managed site for a number of cancer data sets. One thing to note is this is not HCMI specific. To find HCMI models, search for the HCMI CMDC project to find the list of models from the HCMI. Here you can download the aligned reads from the whole genome and exome sequencing and gene expression from the RNA sequencing data. ATCC's website delivers information about the models, is available for purchase. It gives overviews of the HCMI program and emphasizes actual cell culture techniques and information. Each individual model page will give you guidance on the exact complete medium formulations specific to a select model, as well as the subculturing, thawing, and freezing protocols. Protocols exist in various formats, including individual documents on each product page, an organoid cell culture guide, and a publication on organoid culture from ATCC R&D scientists. Again, I want to note that from each model product page, you can find a direct link to the HCMI searchable catalog and genomic data common page. So now I just want to give you an overview of where we are today with the HCMI models. We have launched over 240 models, including 155 organoids. These include various stages of disease that come over a variety of tissue types. We are planning to continue to launch more models over the next several months, so stay tuned to get information for future models. And here I want to go into more detail on what ATCC offers in the way of culture support for organoids. The basic protocols used for subculturing, thawing, and freezing are largely constant for most organoid models. They don't really differ from one model to the next, and we offer these protocols on our website. The biggest difference between each model will be the exact media formulations. There are different formulations that cover the current organoid models offered. They all use a base media with standard components, but they differ in that most use conditioned media and different assortments of recombinant proteins and small molecules. On that note, ATCC offers Organoid Growth Kits that supports the formulations. These organoid growth kits consist of small molecules and recombinant proteins that I just mentioned. Packaging them in one kit greatly simplifies preparation of the complete media growth. These kits will be single-use lyophilized aliquots that require no real formula calculations. They're designed to be added directly to the base and conditioned media. The growth kits also alleviate the need to buy and store bulk reagent components from different vendors. Lastly, ATCC uses these same kits in the manufacturing of our own organoid models. On that note, we have an educational video on how to prepare organoid growth media presented by Stefan Friend and James Clinton.
Hi, my name is James Clinton. I am a scientist at ATCC. Primary tissue-derived organoids are a three-dimensional in vitro cell culture model that permits the long-term growth of patient-derived human tumors that can be passaged, expanded, and cryopreserved for later use. ATCC is supporting the Human Cancer Models Initiative by making their collection of models, including organoids, available to the research community, and by providing tools, reagents, and training to help scientists utilize these models in their research. This video is intended to help tackle how to prepare organoid culture media utilizing prepackaged kits made by ATCC. These kits, when combined with other reagents, are designed to facilitate the creation of a complete growth formulation for various primary tissue-derived organoids, including those developed from the Human Cancer Models Initiative. Kits should be stored in a non-frost-free freezer at -20°C or below prior to use. Remove the kit from storage and transfer it to a biosafety cabinet. Refer to the specific media formulation instructions for any additional reagents you may need and the precise volumes necessary. These reagents should be thawed in advance if needed. Start by preparing any components not provided by the kit, including basal media, buffer agents, glutamine, and other supplements. Because the organoid growth kit components are provided as powders, Briefly centrifuge the vials to ensure that the contents of the tubes are not stuck to the walls or caps of the vials. Following the instructions provided, reconstitute the individual components in the correct buffer. Typically, this is either in media or DMSO. Mix thoroughly by gently pipetting up and down. Let the vials incubate for 15 minutes at room temperature. Some components may be difficult to reconstitute. To help the material enter solution, incubate the vial in a 37 degrees centigrade water bath for 10 to 15 minutes and then briefly vortex. Depending on the media formulation, additional components may be required, such as conditioned media derived from specific cell lines designed to overexpress organoid culture relevant growth factors. After the reconstituted kit components have been incubated for 15 minutes, add them to the previously supplemented basal media. Add any additional reagents indicated in the media formulation, such as conditioned media. Conditioned media provides a highly active source of some recombinant proteins that can be essential for organoid culture. The final growth media can be vacuum filtered, such as by using a 0.22 micron bottle top vacuum filter. An adhesive sticker is supplied with the kit and can be used to label the final bottle containing the prepared growth media. Once prepared, store the growth media at 4 degrees centigrade and avoid extended light exposure. We do not recommend freezing the complete growth media. This was an overview of ATCC's Organoid Media Growth Kits. For other information on working with organoids, check out the ATCC website for our Organoid Culture Guide, media formulations, and a video protocol that guides you through the critical steps to successfully culture primary tissue-derived organoids in your laboratory.
This concludes our presentation on HCMI organoid models and culture. To recap, support for these exists in the way of information support on clinical and genomic data via NCI's HCMI searchable catalog and the GDC data portal, all protocols and organoid culture media formulations, growth kits to make organoid culture easier. ATCC offers 240 HCMI models and will continue to launch new models in the upcoming months. For more information, please check the ATCC website to download the Organoid Culture Guide and feel free to watch the video again at your leisure. And thank you, Dr. Shapiro, for setting up this webinar. We can now go to the Q&A portion of this presentation.
Well, thank you, James and Steve, for the excellent presentation. In just a few moments, we will begin our Q&A session. Please remember to use the chat function available through the webinar program to submit your questions. The session will be documented along with the recorded webinar presentation on the ATCC website at www.atcc.org. So we've already had several questions come in. Let's go ahead and jump in with the first one. Maybe I'll send this one to Steve. Can the organoid growth kits be used for other organoid models that aren't offered by ATCC?
That's a good question. These organoid kits were specifically made for our organoid models, which are, again, patient-derived. from tumors. They may work in other types of organoids. We cannot validate that or verify that. There is commonality among different types of organoids, so it is a possibility. But again, these were specifically made from formulations that were known to match our models. So again, they're made specifically for our models, and we can't really verify if it would work in others.
All right, good. Now, James, this question's a bit more technical in nature. I guess I'll send this one your way. Can I substitute R-spondin or Wnt instead of conditioned media?
I assume the question is, can you substitute with recombinant R-spondin and Wnt instead of the conditioned media? And yeah, I think we get that question a lot. I think it is definitely possible, sort of similar to what Steve just said. That's not something we've thoroughly tested and validated. We've found that often the recombinant proteins will have a lot lower activity in culture, and thus they may not be quite as good as the conditioned media source. But it very well could vary model to model. So I wouldn't say you can't do it, but we cannot guarantee that'll work certainly as well, and it has not worked as well in our hands. Obviously, there's lot differences with recombinant proteins. There's multiple vendors and all of that. So there's a lot of variables there that we just have not we've not fully explored. So you can do it, although it is not our recommendation.
Gotcha. And is each kit specific to a tissue type? And I can open that up to either Steve or James to answer.
I can answer that, and I'll say the answer is no. There is some overlap. Some kits are actually used with multiple tissues. It's not tissue-specific media formulations. It's slightly more complicated than that, unfortunately. So I would encourage you to look at the model-specific information that we provide for any specific organoid model, specific part number, and see what media formulation and what kit it recommends.
Okay, that makes sense. Now, this is just a general question about the kit itself. What is the volume of media made by a kit?
Sure, I can answer that. A complete kit is used for 250 mils of media. And again, the complete formulation is on our website. So the base media would be advanced DMEM, which comes in a 500 ml bottle. So you basically split that in one kit. Again, each kit, regardless of the type, is used with exactly 250 mils of final volume of media.
All right, excellent. Now, the kit comes with several vials of other components. Why does one or more of the kit component vials look empty?
Oh, okay, that's a good question. So the thing about these kits that make them so useful is a lot of these reagents are needed in very, very small amounts. And so they're lyophilized, and quite frankly, the volume is so small you literally can't see it. And that's what makes these kits so good is if you were to try to make those volumes on your own, you would have to buy them in bulk and then radically dilute them and have a ton of material you may never use. So these are meant to be, they're in small volume because they're meant to be used as one-offs. Again, each kit, 250 mils, and again, very small volumes of reagents are in the kits.
Okay. Do you have any suggestions to make sure that you get all of the powder or all whatever's in the vial in solution?
Sure, I'll let James answer that question since he spends more time at the bench than me. He could probably give you better tips.
Sure. So it's important to follow the instructions we provide with the kit. When dealing with really small masses of lyophilized proteins or even just powders, spinning the tube is a good first thing to always do to get the any material that might be stuck to the cap, for example, down into the bottom of the vial. The next thing to do is make sure you're using an appropriate volume, and don't just volume of a buffer to reconstitute whatever's in the vial. You can dispense it along the walls to kind of wash the walls a little bit, and just generally make sure you're kind of coating that interior surface. So I assure you we're not sending out vials that have nothing in them, but it is really important to make sure you're reconstituting them in a way that gets the material that's in there, be it even if it looks like it's empty, there's like a film at the bottom. So it's just important to follow the instructions we provide when reconstituting the kit components.
Okay. And I think this point was made pretty clear in the instructional video, but are the kits alone sufficient to make media? And if not, what else is needed?
Yes, I can answer that. The kits are not all of the components you need to make the complete media. It all comes. All of our kits use advanced DMEM and other things like B27 and so forth. Everything in L-glutamine. Those are reagents that are in larger volumes, like 10 mils, 5 mils, things of that nature. And those reagents are listed on the protocols for each model on the website. So, fortunately, though, we don't offer the complete solution, but we offer the harder to obtain reagents and recombinant proteins and so forth. And so you would need our kits, advanced DMEM, and maybe a couple of other reagents, and the conditioned media as well, because the conditioned media is made separately also at this point.
Okay, excellent, excellent. All right, now this asker would like to know what's happening. They added the buffer to the bottle or the vial of N-acetylcysteine, but it's not dissolving. So what's happening? Is this expected? And maybe what to do about it?
Sure, I can try to address that. So N-acetylcysteine can be difficult to get back into solution. Again, in our kit instructions, and I believe it even does that in some of the videos as well, we recommend, or what can help get into solution is to warm it in a 37-degree water bath for a period of time. Also, vortexing it repeatedly can help it enter solution. It's not uncommon that sometimes it can be difficult, but it will go into solution with a little bit of that.
Okay. Now, this is just a general question about culturing organoids. How often should the media be changed? Probably a good one for Jim.
Yeah, I can take that one too. Like as I think with all cell culture, it sort of depends on the specific model that you're growing. So it's same with cell lines. The models that grow faster will need their model replenished more frequently. We in general recommend, as a general rule, we recommend changing every 2 days. We found that to be very effective in supporting the growth of the vast majority of models very well. That would be my general recommendation. There may be model-specific guidance that says otherwise. Maybe not more frequently, but maybe even sometimes less frequently can actually be beneficial. But again, it's highly model dependent. So my general response is every 2 days. Start with that. If you're seeing, obviously that the media is quickly turning very acidic. The phenol red indicator is turning bright yellow. You probably consider changing the media more often. But yeah, start with every 2 days as a general guidance.
All right, good. Now, can other commercially available medium be used for the HCMI organoids that ATCC provides?
I can comment on that. Maybe James could add to it. It's kind of a similar answer to what I gave the other question about could our kits be used with other models. It certainly is possible that other on-the-shelf media can be used with these organoids. Again, we have not investigated this, I don't think, very much. So that would be sort of used at your own risk kind of a thing. And I don't know, maybe James can add on to that if he has any more updated knowledge on that.
Well, I mean, that's basically what I would say as well. I mean, I would not be surprised if there are other media formulations out there, either commercial or otherwise, that will allow the models to grow. The question is, well, one, will they maintain the same phenotype? We don't know. They're certainly not, if you're not using essentially the kits that we provide, you are not using the same formulation that the models are used to during manufacturing, so you're deviating from that. Again, that could have some sort of phenotype or phenotype impact. So again, we recommend sticking with the kits because that's how the models were kind of derived and manufactured. Any deviation from that comes with some amount of risk, and it's ultimately up to you for the end user then to kind of validate that in their particular application. Maybe it'll make a difference, maybe it won't, but we certainly can't tell you what impact it will have because we have not done every single possible formulation comparison via homebrew or commercial with any number of the 100 and 100 plus organoid models we currently have.
All right, good answer. Good answer. All right, we've had a bunch more questions come in, and this question I'm about to ask is around the characterization of the models themselves. So how well characterized are the models, and maybe is there some differentiator that you could mention between our models and what other models there might be out there? I know that was very vague and sweeping, but maybe if there's a couple things that you could comment around that.
Sure. I mean, I can take a stab at it. I mean, how well characterized are these models? I would say vary, but ultimately it depends on what type of characterization you care about. These are all derived from the HCMI, and so they are subject to a variety of molecular characterizations. So there's RNA-seq, there's whole genome sequencing, there's whole exome sequencing. If you consider that important levels of characterization, it's great, and I think many people do. If you're looking for other types of characterization, like a lot of histology, for example, unfortunately, no, we don't have that. If the question is sort of, why would you switch? I would sort of argue that's not a characterization level question. It's, if you are looking for a specific indication that maybe you can't source yourself, then that's an option from ATCC. Like we have 18 different. We have organoids from 18 different primary tissue sites. So maybe there's a tissue there that you're not able to source otherwise, or maybe it's you only have an N of 1 or 2, and you'd like to increase that to say 40 or 50. Then we have a large collection that can increase your sort of sample size. Maybe you're looking for a specific combination of mutations. Then again, it's not really a switching situation. It's you can mine our database of the models that have been sequenced and say I'm interested in these with mutations 1, 2, or 3, or whatever combination it might be. It might also be because of just the demographic information. So again, we have clinical information from the patients that the models are derived from. So maybe you're looking for a specific gender between a certain age that did or did not have neoadjuvant treatment. So I would point to all those things as reasons why you should consider organoids and other models from the HCMI and HCC's collection. I'm not going to try to persuade you to move away from models that you already have if they're working well for you and if they're characterized at a level that works for you. That would be my kind of answer to your question.
James, that was a very nice answer. All right. So let's see. Next question. This is probably a quick yes or no. Do we have any lymph node organoid models?
Yeah, I can answer that. Unfortunately, we do not at this point.
Okay. And then let's see. What if an institution is interested in depositing samples to the HCMI? I guess the question is, do you guys have any window into how to go about that? Is that possible?
I'll sort of give a general answer and again let James, because James works very closely with the model developers. He could probably give you more detail. There's certainly, we can certainly give your contact information to someone in the hub or the model developers that we can guide you in a direction to see what the process is of becoming one of the developers. James, do you have any more specific insight on that?
Yeah, I think this is a question for NCI. This is not a ATCC governed program. We do not choose the model developers. NCI would need to, they're the ones who decide what models and who can deposit as part of the HCMI.
All right. So in that case, it's probably on HCMI to ensure HIPAA compliance when sharing genomic sequencing information, correct?
I can try to answer this question. So yeah, so essentially, as part of the HCMI, patients were enrolled and gave consent to provide tissue, which would be used to generate models. As part of that, they gave permission for sequencing and resequencing. The sequencing data that we and the HCMI make available, there's essentially two kinds. There's controlled and uncontrolled. The uncontrolled data, which is essentially the publicly accessible data, is somatic mutations only. Nothing's very low frequency. Essentially, there's no way to track back to an individual patient. There is controlled data that is essentially the complete raw, essentially all the raw data. That requires essentially working with NIH to give, essentially you sign some documents that saying you're going to maintain the patient's privacy, you're going to maintain the data in a proper way, and this is all done via DD-GAP. So this is standardized how they handle sort of patient data like that. Anything other than that is not considered sort of personally identifiable information. So we again do have kind of high-level clinical stuff. You know, we have things like age, primary tumor site. We have some treatment history around therapeutics. We have maybe histology around the tumors and things like that. So, and I will also add that ATCC does not directly provide any of the sequencing data. Right now, this is distributed through NIH and NCI platforms. So they would be the ones. If you have very specific kind of HIPAA-related questions, it would be best to direct them there, not to ATCC. Unfortunately, we cannot answer.
All right. No, that was also a very good answer, James. Thank you. Okay. I think that we kind of hinted at this earlier, but I guess more explicitly, can we use conditioned medium from WRN-3A cells that produce all three components, the Wnt, R-spondin, and noggin, instead of the recommended Wnt3A and the cell line that produces R-spondin only?
So yeah, I can take this one. I mean, again, it's can you? You know, most likely, yes. Certainly, if you look in the literature, a lot of people do use the LRIN cell line to generate conditioned media and supplement it for organoid culture. Again, we do not do that internally. We do not do that for manufacturing. It makes it more difficult to individually manipulate the levels of Wnt, R-spondin, and noggin. In some cases, not all models need, for example, Wnt. So again, it comes down to you can do that, but then you are not following the guidance that we have provided, at least with our models. For other models out there, I'm not going to say one or the other. It would likely support growth if those are vital components as part of the culture.
Yeah, and one thing I'd like to add on that is, so we're talking about the three components, Wnt, R-spondin, and noggin. Our kits do have noggin in it as an individual vial reagent. So if something to take into consideration, I don't know if that would be, if you used the noggin we supplied and that media that makes noggin, is that too much in your, maybe too much in your final growth media? I don't know. I mean, if you did go that route, you could simply not use our vial, but that's just something to take into consideration. Our kits all contain noggin. Again, it's individually vialed, so, and it's on vial, so you could exclude that if you wanted to use our kit in that media. But again, something to just think about, take into consideration. I don't know if James has any opinion about that. The volume of the reagents is pretty specific. So I don't know if that would cause problems.
Okay. Oh, and so the asker actually just followed up with, the problem is that the cell line producing R-spondin, I guess, recommended is no longer available. So
yes, we right. I was going to say we are actually looking, because again, that's not our product. We are sort of looking into alternate sources of that ourselves. I would say when we find possible solutions, we will update our website in our product sheets or the formulation sheets that could possibly give some solutions, because we became aware of that recently ourselves, and we're sort of working on an alternate solution.
Okay. Good. All right. And let's see. This question actually echoes a question that you guys answered earlier. But is there a recommended kit specifically for prostate cancer organoids?
We do not have a kit. We do not have any prostate models in the HCMI collection, so we don't have any kits for that at this moment. So again, we know that prostate cancer models are something that's very highly sought after. We would like to have some too, but we have not received any prostate cancer models from the model developers. So no, unfortunately, we don't have a kit that we have verified will work in those models.
Right. And you would extend that to melanoma and lymph node as well, specific media recommendations, correct?
Correct, yes.
All right. Well, it looks like those are all the questions that have come in. I guess while we're giving last call for other questions, we do have another webinar coming up that you all might be interested in. It's around navigating the HCMI searchable catalog and accessing the HCMI data at NCI's Genomic Data Commons. This is going to be presented by Eva Thompson-Carter, who's the Scientific Program Manager at the NCI Center for Cancer Genomics, as well as Bill Wysocki, the Lead for User and Data Services at the Genomic Data Commons Center for Translational Data Science, and our own James Clinton, who you've heard from today. All right, it doesn't look like we've had any further questions, so at this time we'll conclude our Q&A session. I'd like to thank James and Steve for the excellent presentation, and thank you everyone for attending the webinar. For more information about culturing organoids, please navigate to www.atcc.org/organoids, or for organoid kits, it's the same thing except it's organoid kit at the end. So thank you again for your time today, and have a great day, everyone.